last webinars, we kind of already talked about how the data is made public with the help of ENA, so the European Nucleotide Archive, and the European Genome Phenotype Archive, and how Galaxy can import data, more or less, from these public archives and then process it. We have also seen last week what happens if Galaxy processes data so that you can more or less then push data back to public archives or to GIS8 and so on. And today we would like to highlight how this processing is, so the central part, so the workflows here in this case of the variant calling workflows. So we will have Wolfgang Meyer presenting the latest Galaxy COVID workflows. And after that, we have Barbita from CRG talking about the new Beacon and the Beacon project in general, and how Beacon actually takes data from Galaxy and then displays it nicely so that you have a nice visualization filtering tool on top of the data that has Galaxy has processed. This is a different view on that. So data mobilization aspect. So data is produced, data is put online to the community. We have this data access model, which is, for example, ENA, the analytic workflows, and then the visualization and navigation, which is now and will be talked today, the viral Beacon project. As usual, you can read everything on GitHub. So there's this website where we get all the information and all the workflows for you to consume. You can deploy that on your local Galaxy server, national Galaxy server, or you can use one of the public Galaxy servers like the European Galaxy server, the Australian Galaxy server, or usegalaxy.org, the American Galaxy server. Okay, and with that, I would like to hand over to Wolfgang, which will guide you through the latest about COVID workflows in Galaxy. Wolfgang.
Okay, thank you, Björn, for the introduction. Welcome, everybody. Good afternoon. So what you're seeing on this slide is basically the status of the project when I was talking about it in summer last year in the first round of this webinar series on COVID-19. And what we had at that time is basically a response to the early needs of science during the just emerging pandemic. So we had workflows for SARS-CoV-2 genome assembly and for pre-processing of such data for the assembly. We had workflows for estimating the most recent common ancestor of the virus. So basically these questions: when did this virus emerge? How did it emerge? How different are different isolates from different countries, and so on. We also had already variation analysis workflows. These were mostly tailored to the most common data at that time, which was Illumina whole genome sequencing data. And then we demonstrated how this could be used in another workflow to do some S-spike protein analysis on top of these called variants, and some preliminary evolutionary analysis. So since then, of course, what's needed and done in science and in diagnostics has changed quite dramatically. And I mean, by now we have more than 100,000 assembled genomes of COVID-19 and many more variant sets in the form of consensus sequence data on platforms like GIS8, for example. And people have started, well, they still do Illumina whole genome sequencing, but also, of course, a lot of Illumina Arctic sequencing and a lot of Illumina, Oxford Nanopore Arctic sequencing. And assembly is pretty much of, well, only minor interest at the moment, because, I mean, you have enough reference genomes so that you don't want to start a de novo assembly again. There's not much use for that. So the interests have shifted quite considerably, and we've been following that all the way and prepared a lot of new workflows, modified others to better fit the needs of the community. And if you can just go to the next slide, Björn. Okay, so this is basically what we try to address within variation analysis. So leaving assembly completely out of the picture now, just focusing on variation analysis. These are the protocols that we would like to support. And these are Illumina whole genome sequencing data, still comes in paired and single end form, of course. Then Illumina Arctic data, which is typically paired end, and then Oxford Nanopore Arctic data. And there are quite some challenges um, going on here. So first of all, bioinformatical challenges. So one is the question, just how many different workflows do we need to analyze all the data? How different do these have to be? So does each of these data sources require completely different workflow architecture with different analysis tools, or can they all be analyzed same way? And then, of course, will data be comparable across them? So if I compare a variant list produced from Oxford Nanopore Arctic data and one from Illumina whole genome sequencing, will they have anything in common? Can I really say this variant was present in this sample and absent from the other when the data sources are very different? Then there are usability challenges. Um, First one is, what's the right amount of user configurability? So of course, for certain data sets, you might want to change certain threshold settings for variants, like say the minimum allele frequency at which you would like to call a variant, when you would call it a consensus variant, these kind of things. Users might want to configure that, but on the other hand, if you do this extensively, let users configure like every parameter in the workflow, how comparable will be the data generated by different users and projects. And then what kinds of outputs to generate. So what does a user really want? You can, of course, produce just consensus genomes, like publicly available through GIS8, but then you're throwing away a lot of the data that you actually generated, because a consensus sequence is really just like the most frequent variants that you're really sure about, while there may be many more which are kind of sub-threshold intra host variants, as we call them. So things that are going on inside one infected person where the virus continues to evolve. And you might throw away much of this information by just looking at a consensus genome. But how then to report this more complex true picture? That's another issue. Um, and finally, there are technical challenges, and these have been surprisingly complex, I have to say. So one is scalability, and I was already talking about this last summer, and I said that we are doing quite well on that, and that was actually true. We still improved scalability of workflow scheduling, job scheduling in Galaxy a bit, so everything's faster, smoother, more reliably running now. But then there's also the question, how robust are your workflows or pipelines in general, not just speaking of Galaxy, but of general pipelines developed in research in the face of unusual input data. So the point here is that we're trying to analyze data in a massively parallel way. We're not talking about a handful of data sets. We're not talking about 100 data sets, but we're talking about tens of thousands of data sets, right? And if you have tens of thousands of data sets, there's always something unusual included in your analysis. Like things would be that I would could think of would be really bad input quality. So something where you just cannot align any reads reasonably well to the viral genome. So you produce maybe even an empty BAM file with aligned reads. Um, you could have very unusual variant lists. So first of all, your variant list might be empty occasionally. So you just don't find any variant for this particular viral isolate, maybe because you have low coverage. You could have multiple alternate alleles. That's of course not an issue if all you try to do is build a consensus genome. But if you're going for these lower frequency variants, then of course you could have multiple of them at the same reference position. So some viral copies might change an A to a G and others might change it to a G. So is then your workflow capable of reporting this correctly, or will it just crash because it has two things at the same site? And then another issue are, for example, overlapping indels, because indel calling is never as accurate as single nucleotide variant calling. You might have just slightly different indel boundaries for the same ref allele. And again, yeah, what does your workflow do then? Will it be able to cope with that when trying to report those? And these are many of the, and I mean, it took us like half a year or so to really eliminate all these potential pitfalls because you only see them coming up occasionally with this one outlier data set that produces them, right? So when you analyze your data like we do in batches of, say, 2,000 samples at a time, so for a long time during the development of these workflows, we always had that one outlier that we never saw before in a new set of, say, 2,000 data sets. And then we had to modify the workflows, make really sure that we're catching all potential errors that could emerge. And this is why now that we have analyzed, like, I guess, 100,000 publicly available COVID-19 sequence data sets, we can really say that our workflows are quite robust by now. And so normally everything should just run smoothly. Fingers crossed that this will still be true tomorrow, but I think so. Yeah, we can be pretty sure right now. So next slide. So the number of the question, how many workflows do we need to cover all these use cases? The answer is not 42, but it's 6. So we need 6 workflows to really cover all potential use cases, and they are shown here. So I just took a screenshot from Use Galaxy EU, where we, as Björn said, they're also available on usegalaxy.org and should be available on usegalaxy.org.au, the Australian Galaxy server. But this is from EU because I'm working for the Use Galaxy EU team in Freiburg. So this is the list of workflows, and you can see we have one for calling variants from whole genome sequencing data, single end Illumina data. We have one for paired end data. We have one for ampliconic paired end reads, so Arctic data, paired end. And we have one for Arctic ONT data. And then we have 2 more workflows who are actually not doing analysis work, but are concerned with reporting what is found by the other 4 workflows. One is the consensus construction workflow. So that builds, as the name implies, a consensus sequence from the called variants. And the other one is the reporting workflow, which builds custom tabular reports and plots of the data that are just better to parse for humans than VCF is. So basically the output of the 4 variation analysis flavors is always VCF completely annotated with everything we could find. And then the other 2 workflows are simplified views into that data for better consumption by humans or by public databases. So either consensus FASTA sequence or these tabular reports and plots. And next slide, please. So this is answering the other question, how different do the workflows have to be? Well, for whole genome sequencing data and Arctic data, as long as it's Illumina data, they can be rather similar. So at a very high level, if you just divide the workflows into mapping step, variant calling, and variant annotation steps, you see we can pretty much use the same tools. So we're using BWA-MEM as the reader liner. We're using LowFrec for variant calling, and that has previously been shown in our project to be an excellent choice for use with viral data. So it has exquisite sensitivity, and number of false positive calls is relatively moderate. It gives you accurate statistical control over your variant calls. So LowFrec is a really good choice here. And then variant annotation is done with SNPF, specifically with its COVID-19 release, which brings slightly better, more reliable, robust annotation of COVID-19 variants than the mainline SNPF version. And now when you want to do this with Arctic data, really the only thing you have to implement is primer trimming. that's done with Alva trim in our workflows. And what we're doing on top of that, although it really has a minor impact, is this flagging of tainted amplicons. So what's that? That's basically when you're amplifying um, your different amplicons all across the SARS-CoV-2 genome, and then one of your primer binding sites is affected by a variant in that viral isolate. That primer might actually, since the primers are tied, right? Um, so you have almost every region in the genome covered by two sets of amplicons. And if one of them is, if primer binding in one of them is affected by the mutation, then you might actually get false allele frequency reports. So you might have biases in the allele frequency you're reporting. And so in that case, if that's possible, we're trying to ignore one of the sets of amplicons that is affected by the mutation. That's another trick in there, but otherwise the workflows are relatively comparable. What's really different is the Oxford Nanopore workflow. And this is because we're trying to model here the public Arctic MinION pipeline that the Arctic network is publishing. So we're using Minimap2 as the aligner, we're using Medaka as our variant caller, and then annotation is the same as in the other workflows. But upstream, it's quite different from the other two. That's the high-level overview of the workflows. Next slide, please. And then, of course, under the hood, they're considerably more complex. So this, for example, is the Arctic Illumina workflow. So, of course, you have many auxiliary steps, SAM tools, filter steps, and so on, and filtering steps for variants and reporting of quality of the analysis. But what I showed you before is the general outline. So next slide, please. Um, yeah, what's important is that this complexity of the workflow is not really exposed to the end user. So what we decided for is to give the user at this first level really almost no control over what gets reported. So I discussed this. If you give the user too much freedom, then different analyses tend to get uncomparable because some filtered away certain variants, others didn't. And you really cannot say something's absent here and present there because you don't know what got filtered out. So at these analysis workflow steps, we're really not filtering as much as possible. So we have, these are the input masks that you will be presented with when launching the Illumina whole genome sequencing workflow, the Illumina Arctic workflow, or the ONT Arctic workflow. And you really will only be asked for your input data, the FASTQ Sanger reads, and of course for the reference sequence. And then in case of Arctic, it will also ask you about a primer bed file, and sometimes about a Arctic primer to amplicon assignment. And now, what if you don't have the data? You don't know what it is? Well, it's easy enough because we provide a published history that's shown at the bottom of that slide, called COVID-19 resources. It's available through the EU server too. And basically, the input data besides your FASTQ Sanger reads that these workflows are asking for are all found in that shared history. So you can just grab it there and plug it into your workflow. Next slide, please. Okay, so the strategy is this one. So the 3 or 4, if you count Illumina whole genome sequencing from paired end and single end data as 2 workflows, then it's 4. I'm only showing 3 here. So these 3 analysis workflows are really trying to aggressively call all variants that you reasonably can. So they're they're using very relaxed low frag variant calling settings. So they're really trying to go down to very rare variants with very little evidence. And then the only thing they do is they apply soft filters, meaning they use the VCF info field to flag the very most questionable variants. But pretty much you're getting everything that's somehow callable in these reports. And you'll get VCF completely annotated with SNPF out of that. Then next come, so then the user is basically free, depending on their needs, to choose the downstream workflow they want to employ next. So of course you can take that VCF and work with it downstream in other applications, or you decide to run the reporting workflow, that's the left branch, or the consensus building workflow, that's the right branch on the slide. And then in the reporting workflow, that's where you can now have filters applied, user-specific ones. So all the variants have been called, they are all present in the VCF, but now you can say, I only want I only want to retain variants for reporting where the allele frequency is at least 0.05, for example, that are at least supported by 10 reads in the data, and so on. This is what's shown in that workflow run interface on the left side. It's a bit small, but these options are what you have. It will also take into consideration the soft filters that were applied in the analysis workflows. So these will also get filtered out. And then consensus building works similarly. Again, you can now set thresholds. You can then define what is the minimal allele frequency required to call a variant a consensus variant? The default is 0.8 here. So if 80% of reads say this is this variant, then it's called a consensus variant and incorporated into the FASTA file. You have another threshold, which is basically a lower threshold. And if the variant frequency is below that lower threshold, then it's considered a reference position. So that variant is present at too low a frequency to be called at all. And if it's in between the lower and the upper threshold, then it's a questionable site where it could be either reference or variant. And then an N, an ambiguous nucleotide symbol, gets incorporated into your FASTA. So we're doing masking of questionable positions there. And this is actually also something that you have to do right. So if you just take GIS8 data and you don't know how it was produced, you cannot really know how that consensus sequence was built and whether it was built according to good scientific practices or whatever. Here you have at least documented in your workflow run which threshold settings you used. So that's one of the big advantages of doing it our way. We think that things are properly documented and well, are relatively sophisticated and working with a lot of data sets. Next slide then, please. Okay, so this is the reporting, which I just wanted to focus a little bit on. So if you decide not to do just the consensus sequence, but really look at everything that's there, this is the kind of output you're getting. So in the back, you're seeing this plot, this colorful plot, which actually shows all the samples in the analysis on the y-axis, and it shows genomic positions on the x-axis. And the colors you're seeing in the individual cells of this data are the allele frequency. The allele frequency. So the darker, the higher the allele frequency. Black is an allele frequency of 1, so a fixed variant in that isolate. And this can be used for several things. First of all, it visually groups similar samples together, as you can see. So you can very quickly see what this roughly corresponds, though not always exactly, to next strain clades. You can quickly see what kind of lineages I might have in this data, key positions that are signature mutations, basically, of lineages. They should typically be black here and then shared by many samples. You also see low frequency variants that you would typically not see in consensus sequences in light colors. And then you can also quickly identify like samples that are maybe not such good quality. Like you see this one row where there's a relatively high amount of low frequency variant calls, a lot of pink, light pinkish color in that one row horizontally. Um, this is probably a sample where sequencing quality was just not so great, and so the variant caller picks up a lot of false positive variant calls. So that's a quick way to basically spot an outlier sample which is not good quality. And then the reporting reveals all the essential information from the VCF. It basically pools all samples together. We also have a per sample report, but this is the per variant report. So it pools the information from all samples and tells you which variants were found in any of these. So I just highlighted here. So this is a batch of COG UK data um, from end of November last year, when there was already knowledge about this new emerging UK lineage B117. And so we analyzed that data, and yes, you can see already in this November batch that there were 37 samples. That's the third last column in that report, that carried this famous N501Y mutation, the one of the key mutations of this lineage. Um, you can actually also see that the other key position, the one from the South African and Brazilian variants, is E484K. And you can see that so overall, these were over 1,800 samples in this analysis analyzed together here in this report. You can see that it's totally absent from the British data, but there are actually 6 samples for which an E484Q, so a much more conservative mutation, is picked up. And interesting are also the last 2 columns in this, well, it's cut off on the right because there's much more information than that. But these 2 columns are the minimal and the maximum allele frequency observed in any of the samples. And so you can see that out of the 6 samples, at least one of them had that mutation present at basically consensus levels, so at a allele frequency of zero 0.9 almost. You would actually, if this would be extended to the right, you would see that all the allele frequencies for all samples are reported too, and you can also see which samples were above threshold according to your filter settings and which ones were below, so failed to pass the filters, but still showed some evidence for a variant that a above threshold sample provides evidence for. So this is quite an exhaustive report. What we found by doing that is actually that, luckily for us, there's kind of a sweet spot for the AF threshold that you can configure, um, which applies to both Illumina whole genome sequencing and Arctic data, and that's at 0.05. So at a frequency as low as 0.05, we can still be relatively certain about our variant calls. So our estimated false positive rate there is below 15% for both Arctic and whole genome sequencing data. And you still have a true positive rate, so what you can find, of about 80% at this low range. And if you consider only variants that are like supported by 15% of the reads, so an allele frequency of 0.15, then you really find basically every true variant that there is. You don't miss anything at this level. That's how sensitive the void flows are approximately. Yeah, next slide. The really high level picture, how this now all fits together with Babita's work. So Galaxy, with these different reporting capabilities, has now really turned into a hub for analysis of COVID-19 sequencing data. So you can do pretty much everything one could want with it. So you can produce these VCFs and feed them into downstream variant analysis software of your own choice, or pass them on to providers like the Beacon network. You can build consensus faster sequences and submit them to Next-Strain GISAID, or also to genome surveillance initiatives, which are now popping up in many countries. And this is a screenshot where I just submitted this batch of sequences to Nextclade and let the clades be auto-discovered by the software. So this is just, you take the output, the faster output from Galaxy, and it's directly usable with Nextclade. And then on the left again, the report. So if you really don't want to do anything else with it, you can still look at those reports that Galaxy directly generates and use it for your own data exploration needs. Yeah, that's basically it. So next slide, please, just to summarize what we've done. So by now we really looked at, as I said, around 100,000 samples. So we're usually analyzing our data in huge batches of many hundreds to thousands of samples. And when we produce a history for each batch, so usually we're doing, not always, but we can do this like grouped by the origin of the sample. So you can take batches of COQ-K data, for example, from a certain date, analyze all those together, or group it by technology, whatever you like. Yeah, these are turned then into these 100,000 VCF files, regularly updated. And the question is, of course, how to provide all this information to the community. Next slide, please. Yeah, so we have this analysis capabilities, which can be used for monitoring what's going on with COVID-19 evolution, variant emergence. And the question is really how to share with the community. And this is where Babita and Beacon come into play. So,
Yeah, thanks a lot, Wolfgang. Please ask questions in the chat. We will have time later to answer those questions. But I would like to give now Babita the chance to talk about the viral beacon and how viral beacon displays now those sets.
Thank you, Bjorn. Let me share my screen. Yeah, do you see my screen?
Yes, perfect.
Okay. Thank you, Wolfgang. It was a really nice presentation, and you described your workflow quite nicely. And I take on from here to describe a bit that how we collaborated with the Galaxy team, and they were very helpful, especially in the beginning, to entertain our queries. What I'm going to present here is what we developed as a user interface to see all of this data, especially these raw data coming from Illumina platform, because at the time then there were consensus data available, and you can see, and in for epidemiological studies, it's good. But we were wondering if people might be interested or could be useful to see directly into the raw data. And what Viral Beacon does is to show all of these variants. So you see in the figure there on the genomic sequences, you see this variant. And but before going into detail about Viral Beacon, what I wanted to tell you or introduce to you a bit was about Beacon. So Beacon is because Viral Beacon structure or architecture is based on this beacon, human beacon. This human beacon is an initiative by GA4GH, basically to share this private human genomics data that is not shareable or mostly in hospitals and clinic, for example. This data is generated and it stays there because we don't know how to share the GDPR issues and this federated model. So what it happens is that it doesn't reach to for example researchers where it can be utilized further. So Beacon came with the development of the Beacon was with the idea that you don't tell us what any metadata or any other information, just tell us at this position, do you see this variant or not in your data set? For example, in certain tumor type, do you see this variant at this position? This is the API, it will make a request and then there will be a response. You can return a response, just yes or no. That was the simple Beacon design. And then what we started doing further from there was to evolve it a bit. So, okay, still not sharing much privacy, not hindering with data privacy, but still, can we find out something a bit more? For example, some phenotype features, some annotation of that position. How can I access this data? Can we coordinate, for example, can we reach out with data access committees and ask permission to use this data? So this is what, for example, here you see if you're talking on a WhatsApp chat, you can ask before you could have just asked, okay, do you see this mutation? Yes. but there is much more information. But later, for example, in version 2, what we started working on was that do you see which diseases, for example, this variant is, do you see? And do you have some biosample information? So this is in the development now, and I will not go much more into detail, but but just to give you a flavor, that the type of query, for example, now with version 2 that you can do is to ask all this certain variant based on your genes interest or classification or biosample type, or within this age group to this age group, or within this gender, for example, or in this ethnicity. So all of this is in development. And what we plan to do, or what we are trying to do here with the beacon, is to let's say to put beacon in your data set. For example, you see there are multiple scenarios. There are hospitals with they store data, whatever preferred way they want to store. The data can stay at the place. It won't. You don't have to. The data doesn't need to leave, or you don't need to submit it. But what we can do is to light a beacon, for example, in your own data set. And with a VCF file, for example, we can annotate, or there is a GitHub open access code you can download. You can put your own beacon there, and then these hospitals, for example, or this clinic or institutions, what they can do, if somebody is going to make a query, it can query, ask, communicate among each other, aggregate the response, and then send it back to the user. Okay, we have found this information coming from these places, for example. So this way you don't have to. So it's a federated model. The data doesn't leave your place. You don't show any. You don't compromise with privacy. But however, the researchers can benefit with this information. So before jumping, so this was the idea for Beacon. And then during that time, well, the coronavirus started. And I'm going to jump down viral beacon, but I wanted to make a disclaimer that we are not virus experts. We come from human genomics background, and provided we knew our limitations. So this is why we didn't try to enter into research, for example, with this data. But we are open for collaboration because we have a lot of data, like Galaxy, for example. We have analyzed these data further, cleaned this data. We have, I will show you later also. So we are open for collaboration. It will be happy to collaborate. And if you have ideas, for example, our intention was to provide something useful for the community with the expertise that we have, but not to hinder into the research that is going on. And last but not the least, we wanted to test this Beacon version 2, and the data was already there for SARS, so for SARS-CoV-2, and then it kind of fit in. And this also happened. So when this is the image that you see on your left is where our institute is, PRBB, where we are based. And then during March and April, when the lockdown started, we started getting these kind of pictures of, there's a hospital just next to our building. And then, this is on your right, the image is from where our office is. So this was a makeshift tent for extra to accommodate, it was like a passage. You see, it was created extra for the hospital wing, so that they can accommodate more patients. Below there is a baseball court, so there were temporary beds, for example. So we were kind of shocked, of course, with this image, and we then questioned, can we do something here? And based on what we can do, for example. And this is when we started looking what kind of data we have. Galaxy came into the picture, and we changed our Beacon V2 schema, which was also, it's interesting because we were thinking those times just with the human genomics, for example, and this is the first time then we made our database schema to organism neutral approach. So for example, in the future, it's not just for virus, we can also put Beacon on bacterial data set or other pathogens. So this was also good exercise for us to not think that only human genomics exists. Based on the pipeline, we used ENA, publicly available data set, and it was very nice that ENA was doing this initiative. They were providing this data, different data sets. UK, for example, they opened their data. It was it was good for us, for example, people who are working with publicly available data set. We used raw data. So for example, Illumina data pipeline was coming to us from Galaxy. That time, now I see that Illumina has also developed, Galaxy has also developed their nanopore pipeline. Those time we collaborated with our own biocore team at CRG, and we in collaboration, we analyzed the nanopore data, and consensus data was also, we developed our own pipeline to analyze. And what we did, we assemble all of these data, we put a beacon there on the data, and we developed the interface to visualize this data. Along with, there was a FTP site, so we were uploading all the files in case somebody wants to download and directly start working with the files. Another thing was data harmonization. I just put this slide because it took a lot of effort, manual work, to normalize, harmonize metadata, because we were showing metadata. And you can see there's one example of, the same text could be written as oropharynx, or it could be nasopharyngeal, or so this was manually, it had to be seen and done, and not just for this, for countries, for cities. So this slide is just mainly that when the time is, that you want to put out information as soon as possible, it will be nice that in future we have some good practices to submit data, and to be mindful what we are submitting. For example, we work in EGA, and it's the same for us as well. Metadata that we get is if submitters could be more mindful, it could benefit, for example. These are the data right now currently that Viral Beacon has. We have around 11,000 from Nanopore, Illumina around 76,000, consensus read from ENA we have around 28,000, and this is still, we need to do the data freeze again, so this is till October, and more or less we have more than 70 countries. The interface, I could have done the live demo, but I was not sure, because usually it lags a bit when I'm doing live demo on Zoom. So this is just a screenshot of the interface, how it looks like. First thing, for example, SNP query, you can get more information, for example, for your own, for the position that you're interested in. Their annotation, for example, what is the frequency? Here you'll see what is the frequency of this variant in Illumina dataset, in ONT dataset, and on consensus dataset, to compare. And you can filter out your metadata. For example, if you're looking for only for certain country, you can filter out here. There are different filter options to play with. There are different query types. So SNP query it will give you for certain position. For example, show me all variant, insertion, deletion, SNP at this position. You can check position wise, or there's a region query to say like, I'm interested in only from this region to this. So you can make a query on specific to region, or annotation based. I'm interested in only ORF6 or in S region. So you can look for that. Motif query gives you k-mer searching. For example, you are interested in a certain motif or certain pattern of the sequences. It uses FIMO, and you can extract your much information, as well as amino acid query. Again, you can check for amino acid mutations directly. Region query, this is the one example of how it looks. So you can here quickly see which variants are coming up, and with different data sets. So this here is showing for ONT, Illumina, and consensus. Yeah. Motif query example, for example, you can see which bases are being replaced, and what kind of mutations are there, and how they are changing the sequences. You can also check distribution. There are plots distribution to see what your data looks like. Now, what more you can do? I can only, and I'm sure you have also attended several different talks in this seminar series. For example, last week, Sergey it was a spectacular talk given by him. So I will not go much into detail, just that Viral Beacon can be utilized for sequences for low frequency variants. For example, what I was saying before, like GCAD and NextRain, they have consensus data, and it's good to know what is the common, consensus data is that, for example, here you see the majority, what is the majority variant at that position, and it will give you that. For example, here it is A, so it will hide the low frequency variants. So what that does is, for example, we can miss certain cases like interhost variations. You see in the plot on the left, the low frequency variant will stay on at the low, and only the consensus data will show these red dots. So we can miss this kind of cases. For example, this is with the farm mink variant. If you remember, this mink culling started, and it was noticed, I think, around October, November. Around October, November, this variant started getting noticed in humans as well. But we see that in Illumina data, or the raw data, it was already started in April. We were seeing this occurring in low frequencies. And in consensus below the pink line, you see in October, it's kind of started getting fixed. So these kind of cases, we also see this kind of trend for UK variants. So we are already seeing them in samples coming from April, already starting in low frequency. So I wonder how many of such cases can be detected. We didn't look so much into it, but if somebody is interested, please write to us. We can definitely give you data or collaborate. And another thing we looked, if it makes sense to look into these, and of course there's a plot of these low frequency variants, which is compared to how many of them were synonymous and non-synonymous on different regions, and we see that most of them are non-synonymous mutations. So another thing is to look directly into BAM files, into raw data. So we have JBrowser instance, so you can directly go and look into the raw data. This is also available at this link, so feel free to browse. And it's aggregated in different countries. So if you're interested in certain country samples, so you can just look directly into that. This is another feature in development to trace real-time evolution of the variant from different countries. So this will go in production soon, and we would like to, this is to see like what the cases that I see, so you can see it on the website. There are more information about the pipelines that we use. For example, all of this is in Viral Beacon page. And there's a FTP site. I put the link here to download the data and do your own analysis. We have users so far from around 46 countries. And we see around 150 users active more or less every month. What's the future of Viral Beacon? So this started with just getting into darkness and just like putting like a trial and error thing, but I see that it could be very useful. For example, what Galaxy presented, the pipelines, if in future for not just only for virus but for certain other pathogens, we can light beacon on those data set. We can be prepared. You don't have to wait for a pandemic to light a beacon or to prepare for it. We can do this from way ahead. Another thing could be that is very interesting, that for example, these ACE2 mutations and what is the play dynamic between human genotype variants and the virus mutation. EGA is collaborating with the Host Genetic Initiative. We are getting now slowly there are human genomics data coming in, and it would be nice. We can also integrate this in Viral Beacon, and researchers can get more benefit out of it. Viva studies, virus-wide association studies are coming up. They are getting popular. It would be nice if we can put some visualization. We can get this data and put Beacon on that and make it available to the users. So I see its use. I see that it could be great, this effort for in future. And I thank Galaxy and Galaxy team for providing us data, ENA for making their data open source and publicly available. And with this, also, I would like to say thank you to my team, to EGA team. We put, especially in the beginning when it was in development, we put tireless effort to put something quick, but also, robust. It shouldn't be just, so it took some effort from us, and it was a wonderful team. We all came together working from our home. Some people just got stuck in their countries and with different time zones, and we kind of, well, it was an experience. Thank you, everyone. And please feel free to ask questions. Thank you.
Thanks, Babita. This was wonderful. Especially that, I mean, if Beacon is really able to do that for all kinds of genomes. For example, the Scandinavian countries are spinning up a pandemic research environment currently. I mean, with a focus on COVID currently, but they are already planning ahead and including other diseases. So that would be a nice collaboration, I think, to extend Beacon maybe, yeah, for other diseases like swine flu and so on. So I think that it's a lot of opportunities here to make data more accessible with the Beacon. Just to sum up before we start the questions, so what you have seen in the last 5 seminars is how the European infrastructure and also the worldwide infrastructure has actually spun up a lot of high reliable services like ENA, like beacons, like these Galaxy servers for processing, and all are actually working together to making data for one hand accessible so that you can actually take public data and analyze it compared with your own data, but also that all around the world you get free compute resources to analyze your data no matter where you are. You can process thousands of data set parallel, and in the end you get them also available in some kind of annotation databases like Beacon that really put some sugar on top of that visualize them, that makes it easy to filter all these variants across hundreds, thousands of genomes. And this is all free to use for any scientist on this world. So I guess the key message that we are all kind of working together to make that process very streamlined, from data harvesting to data access to data processing and then data visualization. And a lot of our supporters, a lot of sponsors. So here in Germany, it's a German network of bioinformatic infrastructure that is supporting us. Of course, Elixir. There are a lot of partners in the US, in Australia, that were supporting all these infrastructures. I'm sure in Spain, CRG and so on. I mean, there are all big funding initiatives behind that are supporting these free infrastructures that we would like or that we need to sustain over the next years. So a big thanks to all of that. Yeah, and with that, thanks. If you want to get involved, please join one of these links. All of what you have seen is available as training material, as hands-on material. So if you actually want to learn how to analyze your data, visit our training network. And otherwise, you will get further information on Use Galaxy U about the European Galaxy project. Thanks a lot.